This is the last of the videos from Module 4.1, and here I'm going to talk a little bit about errors that happen constantly when your cells are dividing, especially during DNA synthesis, and what happens when those errors aren't corrected appropriately. So typically in class, what we would do is play this game called Scribblish, in which I provide a message at one end of the lecture hall, it gets passed through each row, and we see what message emerges at the other end. So, for instance, I might give the person sitting on the left side of the room a message that just reads, "A man on a bridge." That person then has to draw a man on a bridge and hand just that picture to their neighbor. So they might draw this. So there's a man on the bridge. That next person then has to look at that picture and draw what they think the message is. So that person writes, "A man crossing a river." Technically correct, but also not equal to the original message. And so that person sends on that message, and their neighbor then draws this picture because they've never seen the original message. So they see this message, they say, "Okay, I can draw that," and they draw it in their head. Man crossing a river looks a little bit different. There's no bridge involved, and so then it gets interpreted, that picture, as man fishing with a pole. So you can see the evolution of the message. Um, and so there are a lot of lessons you can learn from a game like this. One of them is that copies are never perfect. So let's just say that the original message is, "The man hit the car with a bat." Okay, so that message is the original, but as you're copying it, the copies will never be quite right. However, here's the second set of lessons, which is no single error is necessarily catastrophic. So here's error number one. You have the original, "The man hit the car with a bat," and the first error is just a single letter gets screwed up. So "the man hit the car with a bar." That's not a big deal. It's still doing some damage, causing damage to a car. However, then you add a second error. So now instead of "the man hit the car with a bar," it is "the man hit the cat with a bar." And now suddenly we've gone somewhere pretty dark. And so this is the third lesson here, which is some errors are going to have bigger effects than others. And so the other thing that really didn't show up here but would have shown up in person is the fact that if your original message starts with an error, so let's just say that instead of, "the man hit the car with a bat," your original message had so many errors that it started with, "The man hid the tar with a pat." What are you going to make from that? I mean, still errors are going to be added every time you copy it, but you started with something nonsensical, and so now the errors much more easily are going to cause new problems. Okay, so that's the general lesson we're looking at here. And now let's think about the process. So here's the problem we're facing. We have a parent cell. It has a single copy of each chromosome. In this case, there's just one chromosome. So it's a single copy. And somehow it needs to create two daughter cells through cell division, through mitosis, two daughter cells that each have one copy of all the chromosomes. How is it going to do this? Well, we saw previously in Unit 2 and in the first videos for this Unit 4 and Module 4.1 that the first step is to copy the DNA. So synthesize new DNA, that happens during S phase. You go from haploid, that means one copy of each chromosome, to diploid, which is two copies of each chromosome. So that's basically what's happening during S phase. And now that you have two copies, they're both identical, you can split them evenly between the two daughter cells. So we have mitosis. Through mitosis, you form two daughter cells, each with one copy. So that's what's happening. The problem is that every time you do that copying, so in S phase, you're making two from one, every time you do that, you're going to introduce errors. So here's an example. We have the original DNA, and after synthesis, the new strand has some errors. People have quantified this, and we know that for every 100 million nucleotides, there's approximately one error in DNA synthesis. So that sounds pretty good. I mean, your polymerase is doing a kind of magical job here. It's screwing up very little. However, put it into context, we actually have 3 billion nucleotides in the human genome. So that now adds up, and it turns out that without any help, there are going to be about 30 errors every single time your DNA is copied. So every time your skin cells divide, without help, you'll see 30 errors. So that could quickly add up and completely destroy your genome. And the question would be, why doesn't it? So it turns out that actually there are a lot of repair enzymes. So when that DNA polymerase makes a mistake, there are enzymes that come in and double check it. They double check to make sure that the A is matching with the T and the C with the G. They're making sure that errors don't continue. So it's basically like an eraser that comes in and corrects the error. So without repair enzymes, 1 in 100 million is the error rate. So that's about 30 errors every time the cell divides. With those repair enzymes, it's 1 in 10 billion, which, remember, we have about 3 billion, so that's a third of an error for every S phase. It's not nothing, but that sure does sound better than 30 every time your cell divides. So now we start to get into connecting all of that, the errors in DNA synthesis, with the cell cycle. And so what if a mutation changes a protein? I mean, you've already seen examples like this previously in the class where mutations can have no effect, which is probably what's going to happen, or the mutation can actually have a pretty severe effect, and it can change the way that your cell responds to the environment. And so then the question would be, what types of genes, if mutated, might cause cancer? So maybe take a little time to think about that before continuing on. But I am going to continue on, and we're going to talk about cancer, because that's what happens when there are errors in the genes regulating the cell cycle. Cancer is actually not a single disease. It is a collection of hundreds of diseases. What connects all of them is that they all have to do with cell cycle regulation. We can see it's a huge deal. So this is 2019 statistics showing that in the United States, we have almost 2 million new cancer cases a year, 600,000 plus deaths. In Maryland alone, over 30,000 new cases. This is a big deal. It's one of the leading causes of death and disability, or at least huge impacts on a person and their family's lives in the world. So what is it? Again, it's a genetic disease, so it's a change to genes, and specifically, it's a change to genes that help to regulate the cell cycle. So here's our pretty picture of the cell cycle, but it's been distorted because we've had many mutations that have now taken away the control of the cell cycle. So one thing to think about is what happens if we mutate or we remove one of the checkpoints in the cell cycle. If you take a look at this graphic, it's pretty useful for this kind of thought process. If you mutated the G2 checkpoint, for instance, your cells would continue, they'd go through a little bit of mitosis, but then they'd hit the M checkpoint and they'd stop. So it turns out that mutating a single checkpoint isn't really a huge deal. However, they can add up. And so now if you mutate all of the checkpoints, what you end up with is out of control cell division. So in this case, on the left, you see normal cell division. We've got all the normal controls in place. I talked about those in the last video. On the right, the controls are gone. So all of the controls are gone. There's going to be unregulated division of these cells. And in fact, as they are dividing over and over again, you might have more errors that lead to the division happening even faster. So now they're moving faster and they're unregulated. So what normally happens there is you actually have yet another set of checkpoints. And if the cell, typically it would fail at a checkpoint. And so when it fails, it will go into a form of programmed cell death. So basically, if it can't repair an error, it will go ahead and die. And what does that normally look like? The most classic example of this is something called apoptosis. So this is a form of programmed cell death. It's a feature of all multicellular life. And the bottom line is that the cell is going to sacrifice itself for the good of the organism. If you're looking at this under the microscope, you'll see 4 basic steps. The first thing is that the cell will actually look different. It will look a little darker than the cells surrounding it. That's the 6th cell in the center. Next up, we have shrinkage, so it shrinks. The chromatin also condenses. Then the membrane, it basically looks like it's boiling, so it starts blebbing, and the nucleus actually starts falling apart into pieces. And then finally, the cell just starts breaking apart into all these little things that look like bubbles. And the cells that are still healthy eat up the bubbles by, or the little pieces of cell actually, by phagocytosis, and they reuse all the parts. That's the normal situation. However, another prerequisite for cancer would be that those processes also get destroyed. So not only is there too much cell division in cancer, but there's also too little cell death. So normally, again, the cell would be taken out, the cell would die if it's not functioning properly. But in this case, here's P53. P53 is something that triggers apoptosis. And so when it is mutated, when it's not functional, no more apoptosis. And now not only are the cells dividing uncontrollably, they just won't die. And so we can look at this as analogies. Too much cell division would be like cells speeding through all of those cell cycle checkpoints without stopping. Too little apoptosis is basically like nobody's watching the checkpoints, and so the cells can just kind of walk right around them. Okay, so we've looked at the 2 sides of what you need to cause cancer. Now let's look at just the fact that mutations just inevitably add up over time. So while a single mutation doesn't cause cancer, there are going to be many. They will just keep happening every time your cells divide. And a lot of mutations need to occur to lead to a malignant cancer. So as they're adding up, that's leading basically toward cancer, but you can have a head start. And the most famous example of this head start is the BRCA1 gene. So this is very much associated with breast cancer and ovarian cancer. People who have certain mutations in the BRCA1 gene are at hugely increased risk for developing cancer. So we'd call it a head start. And it's so much of an increased risk that many people decide to remove their breasts and their ovaries to avoid cancer in the future. This is something that it can appear in pretty young people. I mean, with this mutation, the cancer might actually start to develop in the 20s or the 30s. It's a very aggressive mutation. So you can have a head start. You can also speed up the rate of mutations over what I was showing you before, that 1 in 100 million or whatever it was. And so adding toxins like cigarette smoke, UV light from, say, tanning beds, certain viruses like HPV, and lots of other things, including, let's say, oxygen. Oxygen, it turns out, is actually a mutagen. So this is inevitable. And so then the question is, why wouldn't a mutation in an important cell cycle checkpoint gene necessarily result in cancer? So take a look back through the previous slides, and hopefully you'll come to an answer for this. Additionally, this is a great video from SciShow. I recommend watching it. It really makes the point well what needs to happen for cancer to develop, and it points out the fact that cancer is not a single disease. So what have we learned here? We've looked at the stages of the cell cycle in Module 4.1. Interphase and M phase are the 2 main stages. Within those, there are substages, so G1, G2, S phase, mitosis, and cytokinesis. And now we've looked at the regulation of the cell cycle and what happens when it goes wrong, and that's cancer. It's a disease of the cell cycle, which, lucky for us, requires a lot of mutations. All right, and so that is it for Module 4.1. Hopefully you've learned a little bit more about the cell cycle and how it relates to cancer.